Thank you very much for joining us. My name is Patrick Poklokla, and I'm a current I&I student here at UCL and your chair this afternoon. We're hoping that this taster session will address those course-specific questions you may have, and will help you gain an insight into what's really like to study in our I&I undergraduate program. So this afternoon, our speaker, Dr. Douglas Fink, will introduce you to some of the great science and medicine that you would learn here as a student at the Infection and Immunity BSc here at UCL. Dr. Fink will also provide a summary of the I&I program, and there will be a Q&A session for the second part of the event. This session is being recorded and will be made available on our website following today's event. So we're here to respond to your questions. And please share these throughout this webinar using the Q&A function on Zoom. Now to introduce our speakers, we have Dr. Douglas Fink. Dr. Fink is a clinician scientist working between UCL and the Royal Free London NHS Foundation Trust. He also works with the Nigerian Institute of Medical Research in Lagos, Nigeria. We also have Meriem Baduni. Meriem is currently studying on the I&I program, just like I am, and is here to chair the Q&A session at this event. She'll be able to answer any questions that you may have about the course or the student experience. Over to you, Dr. Fink. Thank you.
Thank you, Patrick. Great job. I'm just going to share my slides. Super. Can you just tell me that you can see those okay? Right, go into presenter mode. That's okay to see, Patrick. Thumbs up. Excellent. Great. Well, welcome everybody. Yes, as Patrick says, I'm going to do an introduction lecture, mini lecture. So I'll give you a summary of what that entails. I'm the program director for the intercalated and also the non-intercalated BSc in our division, which is Infection Immunity UCL. Presumably, through the wonders of the internet, there are people joining us from all over the UK, maybe even all over the world. So I thought I'd give you a sort of visible sense of actually what UCL looks like. So here is the quad you can see with its characteristic steeple there, and then the cruciform building, which again is in the shape of the X. And we're embedded in this kind of very rich academic, but also clinical landscape in the centre of London, really, really very central. And just here you can see the spearmint corner of University College London Hospital. So the very integrated academic and then sort of clinical space. In terms of the content we're going to go through, I want to give you a taster of the science, so just a sense of what actually a lecture might look like. Then obviously we're going to go through the actual nuts and bolts of what this degree looks like. Importantly, making lots of time for you to ask questions, but also to get some sense from Patrick and Meriem what it's actually like to study. So our division, Infection Immunity, as the name suggests, is concerned really about how infections, pathogens, infectious agents interact with our hosts. And I'm going to principally focus on viruses, which is my particular research interest. And some of this material will be familiar to Patrick and Meriem, I'm sure. So if you break down really viruses into their most four essential components, ultimately they are a genome, which just means sequence of genes, genetic material, and a container for that precious cargo. Ultimately, unless they find their way into a cell, they are completely inert. They're entirely dependent on human cells or an equivalent cell to turn them into a virus factory. So this tiny, invisible, simple structure can somehow harness almost any human cell to turn it into a factory for making many millions of virus particles. And we're really interested to understand how that works and how that potentially leads to disease. When we talk about viruses, we slightly anthropomorphize them. We tend to give them slightly human qualities. So we talk about viruses, even though they're inert, in terms of their life cycle. And the life cycle for HIV, human immunodeficiency virus, is shown here, just as an example of what a life cycle for viruses look like. Fundamentally, as I said, to be able to replicate, they have to be able to engage in the cell and use its machinery So first of all, of course, they need to enter the cell. The surface of the virus fuses with the surface of the cell, ejecting its precious cargo. And this container, sometimes we call capsid, is what encloses the genome, converts itself from RNA to DNA, and then it does this remarkable thing after replication called integration. HIV, when it infects you, quite literally becomes part of the host, literally inserts its DNA into your DNA, and then uses the machinery to reproduce itself. So then it expresses genes, which builds the proteins, and then those form new parts called the bud off, and infection continues. And really, viruses are profoundly, carefully evolved to do precisely that in lots of different settings. This is just HIV, but ultimately, all viruses really have to go through these key steps to be able to be successful. Of course, because viruses tend to damage cells, cells have evolved incredibly nuanced and multi-layered systems to detect and then also eliminate viruses to avoid harm. And those are sort of symbolized here by these triangles. One key defense system is something called interferon. And as the name implies, these interfere with virus replication. What's amazing about interferon is it's basically like an emergency signal. So the host cell detects that there's something foreign, which is typically virus RNA or DNA or a protein. It presses an emergency button, which is the interferon button. That creates a protein called interferon. And then the amazing thing is that interferon activates many, many other different genes. And that leads to lots of different mechanisms for shutting the virus down, which are symbolized here by these little yellow triangles. So it's this amazing amplification and early warning system. It also has a cool name. So if we imagine that in a more complex system, because of course, humans are made of many cells put together, if you have a virus, in this case, again, HIV, infecting a cell, and the nucleus here is symbolized by this sort of oval structure in the middle, and there are 8 cells obviously here next to each other, your cell recognizes the virus and sets off the interferon alarm. The clever thing about interferon is it can then activate not just its own cell, but neighboring cells, which is when we know we've got an effective alarm system. So as symbolized here by the change in the color of the cells, the 4 on the left, that have been turned on by interferon, this is where genes and proteins that we call the antiviral state have been switched on. And these are really effective at stopping viruses from establishing infection, as we showed with the previous slide with those little yellow triangles. And what we have is this eternal conflict, this battle going on constantly, that we call evolution and counter-evolution, where a virus and an immune system are constantly trying to outdo each other. So viruses get into cells, cells recognize and respond, come up with ways to get rid of them, but then the counter-evolution of the virus is to say, okay, don't worry about that. I'm going to use an escape mechanism, an evasion mechanism, or I'm going to deploy my own defenses to turn off your defenses, sort of counter weapon. What does that actually look like, though? Conceptually, these are interesting things, but actually, what does it mean in practice? How does this correlate to things that occur and affect us day to day? So this is Lucy and Lorena, who are old colleagues of mine at UCL, who've now gone on to be successful independent academics in their own right. And this was in 2020, 2021, so in the peak of the COVID pandemic. And when you're working with these viruses at that time, before there was a vaccine, it was considered very high security. So we had to wear special equipment and use these special hoods that suck out air. And what they were trying to do was essentially, rather than HIV in that picture we showed you, they were trying to see what happens in terms of interferon when you put these viruses onto the cells that they'd evolved to infect, which in the case of SARS-CoV-2, of course, is epithelial cells and human airway cells. And on the right, you can basically see the microscope slides of what Lorena and Lucy are doing in that hood. They've put a large amount of virus onto these plates of human airway cells that they put in a Petri dish. And after 5 hours, you're looking for evidence of virus replication. What's clever is that they've engineered the virus in the lab such that you can recognize it to express something called GFP, so a green protein that's easy to detect under the microscope. So if you see green, that means that there is a virus in that cell because they've carefully created a virus in the laboratory that helps us to see it and helps us to measure it. So at 5 hours, you can see there isn't really much going on. 10 hours, some of the cells are getting infected, and by 24 hours, most of the cells are expressing this protein. So that implies that most of those cells are infected with SARS-CoV-2, the cause of COVID, of course. Okay. So why was this important? Again, it feels surreal even to still be talking about it, but obviously this had a devastating impact on how we went about our lives. And you might remember in the Christmas 2020 and also 2021, we were going in and out of lockdowns because the virus was continually evolving around our immune responses, including even the vaccine, and finding its way across households, across the city. And the nomenclature we use for a virus that evolves is usually a variant, but some people were calling them mutants. But we didn't really understand why these new viruses were so effective at transmitting, even more effective than the original virus that was circulating in 2020. And in the Petri dish, you can measure it to show why, or to demonstrate how this virus is better at replicating, the one that cancelled Christmas at the end of 2020, 2021. The original coronavirus here is shown in the light blue bars. So what Lorena and Lucy have done have put a certain amount of virus on those cells. And then at these different time points on the x-axis here, they've measured how much green there is. And basically with the original coronavirus, probably you didn't see much going on until about 72 hours. You can see that the blue bars sort of wobble around quite a low level and they jump up, implying that there's increase in replication at that time, with the level of virus being measured in genome copies on the y-axis. What was interesting about the alpha variant, which was this mutant that cancelled Christmas, was that actually it started replicating much earlier. So it's able to jump up really from about 24 hours, so nearly 2 days before. So that implies that while the original virus would land in somebody's nose, start replicating, and then maybe infect someone 3 days later, actually the new virus was able to do that much quicker. Therefore, in the same period of time, could transmit to many more people. Also of note, of course, is that the new virus, the sort of red-brown color, gets to much higher levels. Although it doesn't look like a big difference on this graph, the spaces between the notches on the y-axis here are a log difference, so 10 times more. So there was nearly 10 or certainly 5 to 10 times more virus, despite using the same amount of virus at the beginning of the experiment. So it moved much quicker and created much more infection. But why? Why on earth did it do that? And it turns out probably that it was because it was able to more effectively counteract the effects of interferon. So the hypothesis was that is this new virus much better at evading or counteracting the immune system and therefore able to spread more quickly at a higher level. So if you measure interferon, as I said, you can measure it as a gene or you can measure it as a protein. Here they've measured it as a gene expression. So RNA is the product of genes. And you can see at 2 different doses, the original coronavirus had really reasonably high levels. So compared to uninfected cells, there was 40 to 60 times more interferon as this key antiviral response. What was interesting was that the new virus, which was much more effective at spreading, absolutely did not create as much interferon, far, far less. And that's shown here at 3 doses on the x-axis there. So clearly this new virus, which has been circulating across the world, has adapted and it's learned how to evade the immune system. And as a consequence, it's been much more effective at transmitting. And this is the paper that showed that. You can see the figure that I've just shown you. So Nature is probably the most influential science journal, really where massive breakthroughs are published. And you can see Lucy was the first author here. And so what we try to do in this in the module or in the course that you're going to come to is actually try and align our teaching about the basic principles of virology and immunology at the frontier of discovery science. So what you'll learn really is what's being discovered almost in real time. And that's a real privilege and exciting place to be. In terms of, again, why this was important, really it helped us understand what the virus was doing as it created many new variants. So on the left here is a figure from a database that basically shows you over time what proportion of the different variants were responsible for the amount of infection in the world. And basically you can see over time that they get replaced by new variants. And probably part of that, as the graph on the right shows, is that each variant is basically better and better at evading the immune response. On the graph on the right, they've used a different measurement of the immune response called CXCL10, which is a protein that measures essentially your innate immune response. And you can see the VIC IC19s, which were the original virus, have much more activation of immunity than the ones on the right, which were the new variants. And that was important because it basically was able to tell the government, listen, this new virus is a big problem and really need to think about how we can stop it. And then fortunately, the vaccine came along. The point is really that for you guys, also for Patrick and Meriem, there's lots of opportunity to stop the next pandemic. So I've taken these news clippings from this week, or certainly this last month. So monkeypox has been retitled Mpox, but that is a virus now that's resurgent. There's a new subclass of it, a new strain called Clade 1b that is spreading rapidly and has appeared back in humans in London. Avian flu has emerged again in the UK and is causing deaths in poultry farms. And Ebola continues to be a big problem in Africa. But the point is, all of these viruses, their success depends on their ability to outcompete our immune system. So you cannot really understand a virus or a pathogen without understanding how it interacts with its host. And for all of these viruses, we need to understand how that works and how we can prevent it from causing harm. And there will be many, many more viruses in the future. We average a pandemic every approximately 10 years. At least that's been the median for the last century. So I expect that will continue. But can we then take a virus from the field? So remember that obviously coronavirus came from bats. HIV, for example, came from chimpanzees. But there are many millions of different viruses that are out there, and it's hard to guess which ones are going to be the next COVID or the next HIV. So is there something about this biology that we've learned about how viruses dodge the immune system that can tell us things about the viruses that are hiding in the jungle, that are hiding in the bat caves, that might be hiding just around the corner, so that we can predict that those are the new viruses that we need to worry about? And those are the new viruses, therefore we can prepare vaccines in advance, so that unlike HIV, unlike COVID, we don't have to shut down because we've already got the vaccine ready and we can protect ourselves in the future. And the interface now between laboratory science, where you conduct your actual experiments, and AI, so we often call that bioinformatic computational science or in silico research, the combination of those techniques means that we can move forward much more quickly. And this is a place I expect that will be very interesting to you guys, because you will be entirely fluent in these computational languages in a way that maybe my generation aren't. So that's about it, really. That's the kind of flavor of the science teaching and the science research that goes on at UCL, the kind of thing that you'll have access to on this course. In terms of the nuts and bolts of the BSc, I'm going to walk through that now. So I hope, as was clear from that talk, the foundation of the teaching is the science. The idea is that you come and you work in a state-of-the-art, cutting-edge research department, and you get exposure to people who are actively researching, so you understand how discovery works, you get curious, you think creatively, and ultimately you learn to think like a scientist. So you're less here to learn a list of facts and develop your scientific skills. And those skills are not just holding a pipette or lifting up blood with a pipette gun as shown in this picture. It's thinking and creating. It's a 3-year course, of course, and you do need some foundations. There is certain basic knowledge and skills that you do need to accelerate from your school. The first year is shared with some of the other divisions across UCL, including the medical sciences program, and ultimately to be able to understand how pathogens work, even to understand how immune systems work, you have to have that very solid foundation in how cells and how molecular biology works, and that's essentially what the first year is all about. But we do continually try and frame it in an interesting and sort of relevant perspective, and always in science. Year 2 transitions a bit more. It focuses a bit more on the infection and immunity aspects. And again, now you're developing a few more of the core science skills. You're learning to communicate in a scientific way, so let's present things either in small groups or in bigger groups. It's also learning your analytical skills. That might be improving your statistical skills, but also just how to look at a figure and how to present it. And again, always aligning that with what's going on in the laboratories all around us. And again, the summary of modules is here. As Patrick helpfully mentioned at the beginning of this talk, these slides and the presentation will be on our YouTube channel. Year 3 is when you really start becoming an independent scientist in your own right. So there are core modules, again, which speak very specifically to infection and immunology in a lot more detail. But the key point is it's all about research. So in the final year, key point is that you get to do your own project in the lab of one of our PIs, so one of the principal investigators. So you're properly getting into the lab or into a computational facility, whichever you prefer. You don't have to be in the lab, but you're actually going to have first-hand experience of what it's like to do science. And again, more direct exposure to a workplace full of scientists. There are, as I said, the core modules in the first term, and then for the second term, again, in and around you doing your project, a range of much more detailed and specifically infection immunology related modules. As I said, there's something for everybody. So if you'd rather do cancer, if you'd rather do autoimmunity, if you'd rather do therapeutics, you'll be accounted for. If you'd rather do infection things, it's accounted for. And of course, there's a whole specialist module on the sort of computational aspect, if that's your jam. In terms of outcomes, so what happens to our, who are you at the end of this? Obviously, the idea is you learn some stuff, but it's also important that you kind of develop as an independent, smart human, that you develop the soft skills of just working with people, of being a professional, turning up on time, but also those sort of hard skills of analysis and presentation. And those are priceless, whatever you go on to do in your career. So yes, there is specific knowledge, but actually most of what you'll do is massively transferable across a range of different settings. And that's reflected in what happens to our cohorts when they leave UCL. Many stay on to do postgraduate research or further study, so often transitioning to a master's program, either at UCL or other major universities, or on towards PhD, so postgraduate research. Some people stay on and work in the labs doing research assistants, but essentially a good bunch of people will stay and do science. But equally, there are lots of people who go on and don't stay in academic research, who go on to do medicine, who go on to do other careers allied to science, whether that's in service laboratories or other jobs, pharma, lots of consulting jobs, really anything at all. Lyle, who's pictured at the bottom here on the left, is a science author, so recently published a book and works for New Scientist. So, there's almost infinite potential. And the key point is, unlike a lot of other BSc programs, you'll come out at the end of your 3 years with this very, very transferable skill set and a real sense of what science actually is like, rather than what you can read about in a textbook. Okay. So, in terms of applications, again, there'll be a bit more information that's available from our application team, if that's the right term. But bottom line is the prospectus gives you your general requirements, and you can, of course, come and actually visit UCL. Like I said, it's in a great part of London. It's a very nice campus right in the middle of town. And then there will be open days next year, although I don't think we have the dates, but you can register interest in advance. And that's, again, links at the bottom here. Great. So I think that is basically the key parts that I wanted to talk about. So I think what would be helpful, if you don't mind, Patrick and Meriem, if you're happy to maybe just do a little bit of your own impression of what it's like, and then we can move on to the Q&A, if that's okay. Thanks very much.
Yes, of course. Patrick, would you like to go first? I'll go after you, hopefully.
Okay, so I think my impression of this course is that it's very up to date with sort of the current scientific literature, and that's something I really value about this course, because they're always changing sort of like the topics, the research. So they've started to include sort of therapeutics on CAR T cells, or lectures about Treg cells, which are being used for dealing with autoimmune disease. And that sort of cutting edge research is something I really like, and I value from this course. Yeah.
Okay. For me, overall, I think this course is generally excellent for anyone who genuinely enjoys science, specifically infection immunity, which is the case for me. In terms of the teaching, it is, as Dr. Fink showed, it is really well structured and kind of builds your knowledge gradually from the basics to the more advanced and applied topics. So by the time you reach third year, which is where we are at now, you are really prepared and confident to using everything you learned, both in terms of your lectures and also in practical, which is in labs and so on. I think one of the biggest advantages is also how much practical experience you build throughout this course. So even if, for instance, lab wasn't your greatest skill during your A levels, I think that this course really shapes and makes that experience very different. I wasn't particularly really a fan of lab work during my A levels. However, through the help that you get from year one all the way until year three, and the extra lab sessions and the up-to-date techniques that you practice with people who are in the fields, you end up building a lot of skills, and those are really important later on, which is in your in your thesis and so on, and other things as well. I think, to be fair, and to also address both advantages and disadvantages of the course, I think one thing that I have to say is that this course is very STEM focused and is heavily scientific and translational, which for me is something I particularly enjoy. So if you're not interested in the science side of healthcare and research and understanding where all these primary research is happening for all the diseases, then this course might not be for you. However, if you are someone who finds this interesting, then I guarantee you'll find this extremely rewarding, and it will set you up well for a wide range of scientific careers, like mentioned earlier.
Thanks, guys. Yeah, that's very heartening to hear. So I think, obviously, I can see one question in the Q&A box, and we're very happy to take others. Shall I put this one to you live? Meriem, maybe if you go first, and then Patrick after. So I guess, yeah, I'll just ask you as it is. So what are you hoping to gain personally from this course? And then what are your next steps?
Okay. So personally, I mean, as the name of the course suggests, I have a specific interest for immunology and infection immunity. And so my aim of the course was that to learn a lot about this specific field, and to be up to date with the latest literature that is out there in this specific field. The good thing about the course is that it directly puts you in contact with people that are literally authors of these papers that you see, and are the ones leading the research. And so that was one of the main, things that I wanted to get out of the course. In terms of my goals for the next step, I think the good thing, again, about this course is that it's not very focused on the science. It's also translational, as I said, and you also get a lot of experience on, a lot of lectures learning about the diseases and the pathophysiology of things. And I think that definitely sparked my interest in medicine, and hopefully taking this research to the next step and begin, helping the patients, hopefully. So I think my next step, hopefully, will be to pursue something to do with medicine.
Yes, ma'am. Yeah, Patrick, go for it.
Okay, as for me, I'd say I'm on the flip side of what Meriem went through. So I wasn't actually quite sure what to do, but I knew I enjoyed sort of learning about the immune system and diseases back during A levels. And so I went into this course and I realized how multidisciplinary this sort of subject is. So it branches off into different fields. So health politics, sort of epidemiology, cancer treatment, and also like et cetera, et cetera. And so what I was hoping to gain from this course was a sense of direction, because you get sort of a taste of everything, as niche as this course sounds. And so that's why I valued that. As for direction, again, because I got to sample so many different things, in my second year, I took a statistics module, and I realized that I really enjoyed doing maths again, like, and incorporating, applying that to sort of the scientific field. And so my goals in the future is to become a bioinformatician, and sort of like try and find biomarkers for diagnosing diseases, also correlates of protection, to see if current treatments are effective. And yeah, those are my goals.
Right. Yeah. I mean, again, your treatments are only as good as the ability to use them. So yes, absolutely, you need to be able to test and monitor. And that's a really important point, Patrick. It does sound niche superficially, I guess, infection immunity, but the ability to connect to all aspects of not just biology, but as you say, even sociology to some extent, epidemiology, even politics, I think is a really good point. So it's a really, really broad skill set you get from this course, and the huge amount of flexibility in the modules that you get to choose. Okay, some more questions. Fantastic. So would you say it is more theoretical work or practical? I'll answer maybe first, and then maybe you guys can chip in as well. So I think although there's a sense that when you're reading things at A level, and even as a first year, there's a sense that it is theoretical. What you realize, I think, after a while, is even the process of reading and understanding something yourself, analyzing something, is a practical science skill. So that actually is part of your scientific method. So I think, although it's sometimes easy to separate theory and practice, actually, the whole point of this is, I guess, treating theory as a practice. And beyond that, obviously, there are more technical skills. And as both the guys have highlighted, there's opportunity to acquire incredibly transferable skills. Those general things like presentation, analysis, but actually, Patrick's going to go into a lab and actually be working with bioinformaticians in his final year. Meriem's going to be working with people who are studying the human immune system in autoimmune disease. And, you're right up against the actual nuts and bolts of practice in the lab. So is that a fair summary, Meriem, maybe first?
Yeah, I definitely agree. I think when I started the course, I had expected the practical side of things to be very different from the theoretical, but ultimately you can't really get into the practice unless you've got a good grasp of the theory. And so they kind of play an exchangeable role in that sense. But I'd say that from first year you do get some practical practice alongside your lectures, so that, for instance, the techniques you may see that are being used to study certain diseases or, understand certain viruses and so on, some of those you actually get a chance to do in the lab, and you're, supervised by PhD students. And this goes all the way until year 2. So there are some techniques that we learned in, I think, a specific module called molecular techniques in biology. And those techniques that we did last year are really transferable to this year in my project that I'm, just starting right now, as Dr. Fink mentioned. And yeah, this year is all about projects and, doing labs as well. So yeah, I think it's a mix of both, and that's the good thing about it.
Right. Patrick, fair to say?
Yeah, I completely agree with everything, but I just do want to mention that sort of the relevancy of these experiments increases as you go throughout the year. So the first year is you learn sort of like techniques that you'll come across very often. So ultracentrifugation, in the cardiovascular module, we learned to do ECGs. Whereas in the second year, we do stuff that's more pertinent to like infection and immunity. So cell culture is something that you see all the time. And you also see sort of gel electrophoresis when you're making blots. So when you come across papers, you'll see that very often. Yeah.
Thanks, both. So there's a question asking, is work placement integrated into the course? So I think I can answer that. In a sense, it is a work placement. I think part of the job of the module is to get across clearly that science is a job, it's a career. It's not a hobby or an interest or a subject. It is a career, and that the people that you'll be working with and be learning from are career scientists. But in the sense that you don't go to a commercial organization, of course, there aren't any placements. But yes, your final year, you are spending months of your time directly working in a science environment, in a laboratory, on your own project. So might be, as I said, on a computer, it might be computational science, or it might be in the lab actually doing the manual work that Patrick was alluding to there, Western blots, pipetting, cell culture. But there is physical hands-on experience either way. Another one I could probably answer, because actually they're stacking up quite quickly now. What would an example of a dissertation look like? So dissertation for your BSc is generally 3,500 words, so it's not much beyond what you have done at A level, but usually it's tackling a really specific question. Do either of you have titles yet for your, sorry to put you on the spot, do you have a title for your, I don't expect you to because we don't need it for another 2 weeks, but do you have a title yet, Patrick?
I'm still ironing my details out.
Still
So,
ironing it out, and that'd be the same for Meriem, I'm sure. I mean, essentially, it would be along the lines of, can we use this cohort of patient samples to define a gene signature that diagnoses latent TB, something like that. So something really very specific, but pretty interesting, and you become a bit of an expert, really. And you really only become an expert in the process of doing the experiments, analyzing the data, and then writing it up. That is science. So yeah, dissertation is a really professional thing and important. Good. So we've done that one. Going backwards, this is to you, Meriem. It says, you said that you have completed your A-levels. So how did A-levels help you get into UCL and pursue your passion?
I think that's a question. Just for clarification, I did biology, chemistry, and maths A-level. I think Patrick also did the same, from what I know.
Yeah, exactly the same.
So I think they do lay a really good foundation for the first year. I think for me specifically, because I did these specific A-levels, which are, as you mentioned earlier, required for the specific course, you don't feel, that the gap between your A-levels and the first year of uni is that huge. It kind of really shortens that gap in a sense, and you are, already, the only thing you have to worry about when you get to university, I would say, is just your study techniques and how you study, because you were equipped from your A-levels. And as you're aware, UCL is a really good university. It's one of the Russell Group universities, and so they do require really good A-levels. And I think since I had UCL in mind, that kind of pushed me to perform really well in my A-levels. And yeah.
And I think I said in the talk, yeah, maths is not an essential component, although I think, and again, it opens doors for you downstream. But yeah, chemistry and biology, I think, are useful. What are the career prospects and progress if I do this course? So I spoke about that in the talk, and if you missed that talk, as I said, it'll be on the YouTube channel. Our acolytes from the course go on to do some really cool things. I mean, they're a very successful bunch of people. A lot of people do stay in science, and a lot of people go on to do PhDs, not just in the UK, but abroad, in the States, in Europe, all over the place. So we make really good scientists, but also people leave science. People stay in sort of biology-related things. So people go on, obviously, to do medicine. That's not an uncommon career path. And then obviously people go and do something completely different. We've had people who've gone on to do law conversions and things like that. And I think that just speaks to, one, obviously, how smart our students are, of course, but it speaks also to the breadth of skills that you can acquire. So I think career prospects are really broad, really good basis for science, but also just a general great training. Okay, next one. Do you think it's better to do biomedical science or infection and immunity if I'm not 100% sure yet, if I'm very interested in viruses, pathogens, infections, etc.? Who's best for that one?
I could probably answer that one. Oh,
Okay, Patrick, you go first, and then
You go, yeah.
Okay, so like I said, I wasn't really sure what I wanted to do at first, and so I almost took a gamble with this one. But like, I'm happy that this sort of aligns with my interests. Like in the end, like I found out that I did like it. Although I think I recommend for you, a biomedical science degree would be more like sort of diverse, and so if you're really not sure, then you could do a biomedical science degree and then specialize further into infection and immunity because you will have exposure to those topics.
Yeah, good answer. Yeah, Meriem, yeah.
Okay, for me personally, I think, as you are aware, I'm not really that huge of a fan of viruses and pathogens. So I think the reason why I studied this course is the immunity side. And you can never understand immunity unless you are exposed to, the basic science on viruses and pathogens and, how they hijack the host and things like that. So I think what the good thing about this course is that it gives you the optional modules. So you can tailor the optional modules to your own needs and your own preferences. And if you in advance have a read on, for instance, immunology, immunity, and you find that is very, interesting for you, then I think this is the right course for you. You don't necessarily have to enjoy both aspects. I mean, ultimately, at the end of year 3, you will deviate to either infection or immunity in terms of your lab and what you end up doing.
Yeah, thanks. Again, I think that's really important. Yeah, you don't have to be passionate about pathogens. But I think there's a really strong argument as we discover more about how the human body works that the immune system is essentially the master regulator. There is no part of the human body that doesn't have some function that's dictated by the immune system. And, a good sort of emphasis on that is there are up to sort of 2/3 of cancers now are treated by some form of immunotherapy. If you have dementia, it turns out actually a lot of that is to do with a faulty immune system in your brain. Even if you have trauma, if you have an accident, a lot of your body's response to the accident, to the giant cut on your leg or the fracture on your bones, actually is an immune response. So immunology is interlaced with absolutely everything in really anything to do with human beings and really to do with any mammalian science. So it's completely transferable. And as both of these guys have said, there's so many different modules you can pick from to make sure you feel like you've got the right skills. Okay.
I think I'll add just one more thing.
Go for it, yeah.
Which is, I particularly found this helpful, is that in this course, you've got a variety of assessments. I think that's another huge advantage. So if you find that you don't perform particularly well in a specific type of assessment, for instance, let's say, written exams, then the good news is that you can, you ultimately will have to do written exams, but that doesn't necessarily have to be all your modules. So you can choose modules based on the content and also based on the assessments. And that doesn't come at the risk of your overall grade of the year kind of, going down. And you get to, have a taste of many different assessments, just presentations, courseworks, writing essays and things like that. You can assess what you're best with and what you can potentially, put in a bit of effort so that you can, master it in the future.
And actually, sort of apposite to that, there was a question that said, please, can you say what assessments are like? Is it ongoing coursework or final exams? Is year one part of your final classification? Do you want to, have a go at that, Meriem, and then Patrick, you can add to it.
Yeah, so as I've mentioned, there are different assessments. Of course, you've got the core modules, and those it depends on the assessment. That the good thing is that you can actually see all of these in advance. So if you click on the link that was sent, you would be able to see the modules that you can choose, and you would be able to see the core modules, and within those you'll be able to see the form of assessment, so you kind of get a good idea. But I think for first year, for instance, we had a few multiple choice exams, we had presentations, which I particularly enjoyed, and I think it then led me to, because I particularly enjoyed presentations in the course, it then led me to open a podcast. So and then that helped me particularly in year 2 and then ultimately year 3 as well. And there are many other forms of assessments, such as writing essays and elevator pitches, where you just have 3 minutes to talk about a very interesting topic and you're assessed on that. So yeah, I think it's not necessarily what you're very used to during A-levels, but A-levels do help with that for sure.
Yeah. Patrick, anything you want to add on that?
No,
Assessments?
I agree with everything, but I just want to include that in the first year, you could do some pretty like fun assessment. I say fun, it's subjective.
Yeah.
In for I believe it was the musculoskeletal module, we made our own website to sort of explain how to maximize an athlete's performance. So given that there are no ethical considerations, how would you sort of
Yeah.
maximize their output, in a given sports? So I think
Yeah,
sprinting.
that's a really good example. I think it's quite different from what you're used to university. There are some traditional assessments, but there's modules where you're marked on how you present, how you speak. There are modules that are obviously related to coursework, which is what your dissertation is. But you get lots and lots of support through that process. And like I said, most of it's a bit more creative than other experiences. What kind of career path or jobs? We've kind of spoken about that. So obviously a good portion go on to research careers, both in discovery science, so academia, universities, but also pharmaceutical companies and other commercial spaces. And then people go on to do all sorts of things, really successful journalists, lawyers, patent officers, consultants, really everything. But, we've got a smart bunch who come through. So you'll be inspired by your colleagues, if not by your lecturers, of course. Okay, key question. Patrick first, maybe. What has been the most interesting lab experiment or module for you so far?
Give me a second to think.
The most interesting.
Sorry, this is like, I know it's
You
only
have
from
everything.
last
Mary,
year.
have you got
Yeah.
anything off the top of your head? Anything that kind of stood out?
So in terms of modules, yes, but in terms of
Yeah, go for module. That's fine.
So I mean, in terms of modules, I think I kind of said this earlier. It was last year, particularly, it was the immunology module. And the good thing about it is that you got quite, a variety of lecturers that are really specialized in their fields, and then they come in and talk about specific, titles of lectures and so on. And you are literally asking questions to people that are very, literal experts in their fields, that are leading the research in the field. And some of them are actually also doctors. So they are leading the research and at the same time treating patients. And we got to learn about all sorts of the basics of immunology. And then we led into autoimmune diseases, immunodeficiencies, cancer, and so on. So I think that was a really enjoyable, module for myself.
Thank you. Yeah.
Okay.
Go for it, Patrick. Any thoughts?
Yeah. Okay. So I'd have to pick the statistics module again because in the final examination, we were given a real life data set from like a national survey, and we sort of had to like rifle through it and like try and come up with our own sort of interpretations of the data, or like what kind of relationships are there that we can see. And I thought that was really interesting because I was able to apply that to other papers, and so uh these like graphs and stuff that seemed incomprehensible to me before actually started to make sense, like methodology wise. And so um I was able to read through those papers, and I was also able to scrutinize these papers. Um so there was one paper that I read actually where they were advocating for a vaccine for treating UTIs, but the results were only significant when ignoring the points that had no results, which there were a significant amount of points with no results. And so I thought that was quite dubious. Um, and they also hit the data points in the box plot in earlier figures. And so I checked the declaration of interests, and um, the lead author of the paper was actually holding stocks in the company that's producing these vaccines. And I would have
There you
missed
go.
that if it wasn't for that statistics module.
So
So
I think
I
that's a
thought that
good
was
example. I
interesting.
mean, there's a good example of, well, two things really. One, obviously, is just the process of analysis, right? And then analysis and close reading is relevant whatever you do. If you're going to be a lawyer, a consultant, stay in science, you really need to pay attention to what's in front of you. But also, it's recognizing that science is conducted by human beings, and it's imperfect, and it's essentially a lot of interpretation of results. Results are not true intrinsically. So yeah, I think that's a really interesting point. Thanks both. Are you assigned a personal tutor like other universities? Where do you go for help? I'm going to race through a couple of these because I think we've only got 8 minutes left. Is that correct? Or we've got longer. I think we've got 8 minutes left.
Yeah.
So yes, you do get a personal tutor. To a certain degree, your interaction with a personal tutor depends on your interest in interacting with them. But most of the tutors are pretty responsive and are happy to support you with applications and give you some guidance. I think that's fair. Do GCSEs play a huge role in this course? Do GCSEs, are they important? Yes, I guess, again, it's just foundational knowledge, isn't it? I mean, your A-levels are obviously your A-level equivalents are the things you'll do just before you come to university. But as Meriem was saying, you'll be expanding on those. But yes, having a grip of biology and chemistry is pretty helpful. Gives you at least some confidence. So how much teaching is at Royal Free and how much is at the Cruciform building? Can you give a typical day in year one, e.g. how many lectures per week and what time do they start? So it's pretty spread over the two locations, certainly probably towards the end, year three, just because our division is mostly based now at the Royal Free at the brand new building called the Institute of Immunity and Transplantation. So it's a brand new, beautiful research building, which happens to be on the Royal Free campus. Most people are more based there, I guess, towards the end of the third year. Meriem, do you want to describe what a typical day in year one is, or what a normal week's like?
Okay. Year one, I can try.
If you can remember.
So I think year one, it was really in Royal Free mainly, and then we had specifically what was really interesting, on the topic of personal tutors, is we did get little group sessions which we called small group teachings with professors, and we were able to have discussions on the topic. So those sessions were in the Cruciform, and then the rest of the lectures were mainly in the Royal Free Hospital. Year two, however, it's year two changes, it becomes a mix. So we had a few still at Royal Free, mainly in term one, and then I think for term two, there was a shift towards Cruciform. And then year three, we're still in the Bloomsbury in terms of lectures, but then labs, as you mentioned, we are in Royal Free Hospital. So it's a mix of both, and I mean, both areas are really nice. So
yeah, it's a nice part of London to be in, I have to say, and they're close together. It's easy to travel up, either bus or the tube, or a bike, or a walk, or a run, whatever suits you. Jetpack, whatever you've got access to. Okay, I think that's probably okay for that one. Going back to the top, how does the program integrate real-world lab experience with theory, placements, partnerships? Yeah, I mean, I think we've covered that. I think it's incredibly well integrated, which is unlike a lot of other BScs. In terms of research, you are literally in the research building. You will be working as a scientist essentially in your final year. And a lot of us, like myself, are also clinicians. So although you won't necessarily be in the hospital, you will be often working on samples that have been taken from patients. So it's incredibly well integrated. As I said, we don't tend to integrate with commercial experience. So you won't go and work in a pharma company, but we are able to support you applying to that after the course. Both AMS and BMS allow the student to select modules, pathways to follow their interests. How did you choose this course versus biomed? I think this person might have missed Patrick's discussion on this particularly. Both of you answered this. So I think the key point is that there obviously is a lot of overlap with biomedical sciences. A lot of the modules, particularly at the beginning, are quite similar. But the key emphasis to as you get further along is you end up with this much more, I guess, specialist knowledge in immunology and infection. And as we said, immunology probably is the single most transferable biology specialism. I mean, it's relevant to absolutely all of human biology. So yeah, it's very transferable. And then of course, the key skills that you learn, communication, writing, analysis, these things are relevant to everything. So yeah, you'll be able to get all of your interests covered within this module. Although superficially it might sound very specialist, actually we cover a lot of key things across all of science. Okay, maybe Patrick first and then Meriem. What would you change about the course if you could? What would you change if you could? Of course, you can say nothing at all. It's perfect.
I was going to say that, and I'm not
Of
paid to
course.
say that, by the way. I probably wouldn't change anything about it.
Wow, that's great to hear. Thank you.
As for me, I think in terms of overall, I wouldn't change anything. But personally, for me, I would think it would be really appreciated if there was like, you know, patient placements, things like that. But of course, that might not be the best thing to do.
Yeah, I think that's an interesting point. And obviously, I think in your final year, we try and line people up to do projects that are more obviously translational and medical than obviously some projects which are much more basic science. I think, yeah, I do think it's. I mean, I'm taking a lot of credit for Professor Milne's work on this course. That I mean, he was the inspiration and the program leader for a long time. And this has been developed over a long, long time, this course, and I think he's done an incredible job.
Definitely.
I think if in terms of things that we might change in the future, I think trying to make sure that there's as much direct laboratory or hands-on experience as possible in the third year will be one thing. Trying to minimize lecture time, I think that would be one thing we might change in the final year. But I do think it's a great course. I am paid to say that. Okay. If you could give one piece of advice to me hoping to stand out in this course, I've only got 1 minute left, especially a personal statement, what would it be? I think for personal statements, make sure you get some advice. Talk to your school, talk to your friends, talk to people who've applied to university successfully. But ultimately, just a well-written, clear sense of why you want to do the module, the degree is the way to go, I think. Not necessarily having done direct science, but just explaining why you're interested in science, I think. Okay. What did you mention in your personal statement? I guess we've only got a minute. Very quickly, 20 seconds, Meriem, then 20 seconds, Patrick. What were the key things you think in your personal statement?
Long time ago, but I think one thing that stood out was, just talking about the pandemic and talking about my experience and how I think that was translational to me wanting to learn more about immunity.
It's a personal statement, so it always is better if you can tie it to your own personal experience. Yeah, Patrick?
For me, it was the fact that you could actually see real world results from this research, like doctors use what researchers like discover.
Yeah, and that's a good answer. Okay. Is research essential for personal statement? And how much research would I need to do now in A-levels? I'd say you don't need to do any research. You just have to be able to explain that you've gone a little bit further beyond your A-levels, that you've done that little bit of reading, maybe you have done some work experience, but really just something that conveys your genuine interest. I think that's the key thing. I presume neither of you had done any research lab experience before university, right? That's fair to say. Yeah.
Yeah.
Okay, and then last question. I think we're in our last minute. Do you do much flipped learning? As is that Marmite?
Quite-
Does anyone? Meriem, you
get
might
the
have to
flipped
translate that for
learning.
me.
Yeah, I
Yeah.
get the flipped learning. So in first year, there was a lot of flipped learning where we'd have to watch some of the lectures in advance, and then we'd come over to the lecture and discuss the things that we found out and what we learned with the lecturer, and ask questions and take it to the next level of actually learning about case studies and things like that. So there was some flipped learning first year, and I think that was particularly helpful in grasping all that information from all different, physiology and, learning all about that. But it kind of gets less as you keep going.
Right. Well, I guess that's probably that then. So I will tie this up, I suppose. Patrick, you meant to sign us off, or Meriem? You're meant to sign us off, aren't you, Meriem? I'm going to hand over back.
Yeah, I'll sign us off, no problem.
Yeah, I'm handing back over to you. So
All
thanks
right.
both. Really appreciate it.
Okay, I mean, we're out of time, fortunately. I would have loved that we'd keep this going for a bit longer. However, if you could please provide us with your feedback about today's session by filling out the survey, which will appear in your browser following today's events, it will be greatly appreciated. And then thank you all for your comments and questions, and thank you for the speakers. Thank you for this excellent session, and have a great evening, everyone.
Thanks, everybody.
Thanks, everyone.
Bye for now. Cheers.